So here we're discussing how the cells actually are divided into two basic types. Fundamentally, prokaryotes or eukaryotes. Prokaryotes means and cells which lack nucleus, and eukaryotes means cells that have a nucleus. Cells that lack nucleus will have a DNA lying naked in the cytoplasm and it will include all the bacteria. Eukaryote include all the organisms which have a nucleus, and these include fungi, the unicellular and the multicellular both. Protocotes, again, these are all unicellular eukaryotes. They all have nucleus. For example, the malarial plasmodium, for example, amoeba, euglena, paramecium. These are all the examples of protocotes. Do not confuse protocotes with prokaryotes. Then there are plants and animal cells. All these have one thing in common, that they all have a nucleus with which the DNA is being housed. So, what are the typical features of a prokaryotic cell? There are some structures that are always present, and there are certain structures which are additional present in some prokaryotic cell and not the others. So, inside a prokaryotic cell, there is a cell wall, but the cell wall is not made up of cell. It's made up of a structure or compound called murine, which is a peptidoglycan. Peptidoglycan means it's having a protein or polypeptide with a small carbohydrate chain attached to it. The cell surface membrane is there, the cytoplasm is there, the circular DNA, and the circular DNA is sometimes referred to as chromosome, and this is a naked DNA lying in the cytoplasm. This will carry all the genes that the bacteria is having, or the prokaryotic cell is having to continue its life. There are 70s ribosomes suspended in the cytoplasm. You can refer to the circular DNA and 70s ribosomes as being the common points from mitochondria and the chloroplast. These are the features which are common to both organelles. If you compare this to the prokaryotic cell, because this is a theory that endsymbiont theory states that mitochondria and chloroplast initially were a separate organism, way beyond our timeline. When present day fungi and protocotes and animal and plant cells were not there, and at that time, when the prokaryotic cells such as bacteria was living. Similarly, unicellular structures such as mitochondria and chloroplast might already be there, but the environment changed, and with the change in environment, the mitochondria and the chloroplast had to seek shelter into a particular cell. So they took shelter into the eukaryotic cell, and since then, evolution took a huge or a giant jump. The eukaryotic cell got the ability to start producing ATPs in large number, and the photosynthetic cells, they also got the ability to synthesize organic compound from sunlight because of chloroplast. So chloroplast and mitochondria took shelter in the eukaryotic cell, and eukaryotic cell gained advantage out of them. This is an endosymbiont theory. Coming back, uh, in the prokaryotic cell, there are some additional structures and sometimes present. For example, flagella might be present. It can induce motility into a particular bacteria. Keep in mind that all bacteria are not motile. Only those that have flagella will be motile. Capsule can be present in the bacteria that will provide additional support. You know, cell wall is already providing support. The capsule can provide additional support. There can be enfolding of the cell membrane within the cytoplasm. These are internal membranes. These are referred to as mesosomes. They give the ability to the prokaryotic cell to carry out photosynthesis as well as nitrogen fixation. So the enzymes for these processes are located on these structures, and these are a bit similar to the cristae of mitochondria. Plasmid is present as small circular DNA, and this plasmid may carry actually is carrying the genes of antibiotic resistance. So bacteria that have plasmid will have antibiotic resistance of a particular type, and the bacteria lacking the plasmid will be easily killed by the antibiotic. So some bacteria can have multiple plasmids at the same time, and the bacteria have the ability to clone a plasmid, to copy its plasmid and pass on the copy to another bacteria which initially lacks the plasmid. So when the plasmid is copied and passed on to another bacteria, both the bacteria will end up having that same antibiotic resistance, and when they will keep on dividing, the entire colony will be having the antibiotic resistance, and that antibiotic will become useless. You cannot use that antibiotic anymore to kill that particular pathogen or the bacteria. How the plasmid is transmitted? This is transmitted through pili. Pili are these extensions from the cell wall. They attach to the next bacteria, and the plasmid's copy. It is transferred through the pili to the next bacteria, which I have just discussed. These features of the table, they represent one thing for the other. The diameter is different. Prokaryotes are very small, 0.5 to 5 micrometer, whereas eukaryotic cell can be 40 micrometer, for example, the plants, and the animal cell is like 10, 20 micrometer. The DNA is circular and lies naked in the cytoplasm. The DNA is linear and is within the nucleus. It is enclosed by two membranes. The DNA is naked. The DNA is having proteins. There are 70s ribosomes and 80s ribosomes are here. No ER present, and ER is present to which ribosomes will be attached. Very few organelles here. Just the organelles that are here are enfolding of the cell membrane and the ribosome, and nothing apart from that. But in eukaryotes, you have several organelles. There are double membrane bound organelles. There are, for example, the nucleus, chloroplast, mitochondria. There are single bound membrane bound organelles, for example, the lysosome, the Golgi, the vacuole. There are no membrane organelles, for example, the ribosome, the centriole, the microtubule, the nucleoli. The cell wall of a prokaryote is made up of murine, a peptidoglycan, which I already discussed with you. Cell wall of a eukaryote can either be of cellulose or lignin in plants, or it could be of chitin, which is a nitrogen-bearing compound in the fungi. And these tables are the repetition of the same. You can just use these for your revision purpose. Viruses are non-cellular entities. We do not classify them as prokaryotic or eukaryotic either, because viruses, they lack any living features when they are outside the cell. They do not have cytoplasm. They do not have protoplasm. Viruses have a protein code called as capsid, and they are very tiny structures. They are so small that they can actually cross the filter paper as well. So they are measured in nanometers. And viruses just cannot be seen with light microscope. They have to be seen only with the electron microscope because the diameter is really, really, really small. So coming back, only a few viruses which are actually in the range of 400 nanometer, only those will be able to seen by the light microscope. So because you know the resolution limit of resolution is 200 nanometers. So viruses need to be transmitted from one host cell to the other host cell, otherwise they cannot survive. So within the host cell, they just inject their nucleic acid and they start to divide and multiply, and they move from one host to another. The viruses can be classified on the basis of the nucleic acid. They contain DNA viruses and RNA viruses. DNA viruses are of two types: the single-stranded DNA virus or double-stranded DNA virus. Or RNA viruses also of two types: RNA single-stranded virus or RNA double-stranded virus. So these are the type classification. Plus, the viruses can be classified on the basis of how what type of organisms do they cause disease to. Lastly, our magnification calculation. You may be asked to calculate the magnification of a particular cell where the actual or real length is given as 80 micrometer. Keep the formula of m is equals to I upon A in mind. Magnification is equals to image upon actual. What you have to do, measure the cell P at its widest region. For example, this measurement comes out to be 60 millimeter. You can convert this 60 millimeter into micrometer by multiplying it with 1000, becomes 60,000 micrometer. You apply the formula, so image upon actual. From the image you got the value of 60,000 micrometer, or the actual value is 80 micrometer already given to you. You just calculate it and you get the answer of 750 times magnified. Another type of question that normally comes in magnification is that the cell is given and then there is a small bar mentioned at the bottom of the diagram and something is mentioned at the top. For example, this black bar is drawn and 6 micrometers mentioned, and you are asked to calculate the magnification of this particular diagram or the cell. You do not take any measurement for the cell. Keep that in mind. No measurement from the cell. What you will do, you will just place your scale or the ruler on this bar and you will measure the width of this bar from this end to this end. For example, I place my measuring ruler here and my answer came to be 36 millimeters. So 36 millimeter is the length of this particular bar. What I'll do, I'll just convert this to micrometer. It becomes 36,000 micrometer. I apply the formula image of an actual. I got the value of 36,000 micrometer from the diagram image, and the actual is already given 6 micrometer. This 6 micrometer is already given to me. I just divided them and I got the answer of 6,000 magnification. The same thing can be done, for example, within the whole cell. They may ask you to just take out the magnification of an organelle, such as the organelle P. So what you'll do is you just measure the organelle P in millimeters along its widest portion. My value came out to be 40 millimeter. I converted it to micrometer by multiplying it with 1000. It's 40,000 micrometer now. I apply the formula m is equals to I upon A. I rearrange the formula A is equals to I upon m because I need to find out the actual size. Magnification, such questions will be given to you, or some one value or the other will be given to you and one will be asked. So in this case, magnification was provided to me. So image upon magnification is equals to actual. So I just rearrange the formula. My value from the image is 40,000 micrometers and my image magnification is 5600. I divide them together and my answer is 7.1 micrometer. So I'll write the 7.1 micrometer. You should know the basic unit conversion and you should know that at times the question just asks you to state the answer in how many significant or it's a particular decimal place. You have to follow that. For example, if the answer was to be reported 7.1 micrometer was to be reported in the nearest whole number, then I should not report it in decimal. I'll have to write the answer as 7 micrometer. So if it says that the answer should be in whole number, so follow the instructions of the questions very carefully. That's it for this chapter.